Hi, everyone. Thanks for joining us today for the webinar brought to you by Microbial Insights on infinitely small forms of life with infinitely large impacts, 20 years of microbial discovery in Great Smoky Mountains National Park. My name is Sam Rosalina. I am Vice President of Applied Innovation. This is my 8th year at Microbial Insights, and I love what I do here, impacting the world in lots of different ways, including environmental remediation, agriculture, corrosion inhibition, and on and on. And I'm excited to be presenting this webinar in conjunction with Dr. Sean O'Connell. Dr. O'Connell is an associate professor at Western Carolina University in the Department of Biology. He teaches classes in general microbiology, microbial ecology, and brewing science. Along with undergraduate and master's students, Dr. O'Connell has been exploring the microbial diversity of Great Smoky Mountains National Park, including species from different forested ecosystems, different streams, caves, and even in elk. Dr. O'Connell earned his PhD in microbiology from Idaho State University in 2002, where he studied the effects of trichloroethylene on groundwater microbial communities deep in the Snake River Plain aquifer. Some of the work he was part of in Idaho and other Western states in the 1990s actually helped to establish the limits of depth and pressure on microbial survivability in the deep subsurface. He helped process unique samples recovered from rock from hundreds of feet below ground that were examined at many labs, including here at Microbial Insights, for microbial and chemical characterization. Dr. O'Connell received a BS in biology and a secondary science teaching license from Johnson State College in Vermont in 1994, and before that he earned an AS in liberal science from Sullivan County Community College in the Catskill Mountains in New York in 1990. He lives in beautiful Sylva, North Carolina, also, as I like to point out, home of the American House Cat Museum, worth visiting, and is married with 2 children, aged 11 and 14. Dr. O'Connell enjoys being outside and doing things like camping, hiking, bird watching, and skiing along with friends and family. We're really excited to have Sean O'Connell here today, and I love the fact that he's had such a long-running history, not only in environmental remediation and bioremediation, which is what Microbial Insights is our bread and butter, but also in working with Microbial Insights as well. So, but I'm also very excited to help with this presentation as well, because it's near and dear to my heart. But before we move forward, I want to remind you all, as always, that this webinar is being recorded and will be available in the resource section on Microbial Insights website by the end of this week. So don't worry, we will be sending out an email with a link and a certificate of attendance following this webinar to those registered. And there are also some included information that we'll provide throughout the presentation itself. So, as I mentioned, this specific webinar is very near and dear to my heart. And the reason for that is that we'll be talking about an organization that I serve as chair of the board for. This organization is called Discover Life in America. And I've been on the board for the past 7 years or so. And it's one of 4 park partners within the park. So Discover Life in America is one of the 4 park partners that are nonprofits that aid the park in different initiatives. So the other park partners are Great Smoky Mountains Institute at Tremont, which is kind of the education arm of the Smokies. There's Smokies Life, which helps with the gift shop and fundraising for the park. And then there's Friends of the Smokies, which does the big, large fundraising. And then Discover Life in America is kind of like the science arm, is how we introduce ourselves, the science arm of the Smokies. And our mission at DLIA is connecting communities, scientists, and discovering, understanding, and conserving the natural world. So our goal is to be that bridge between community scientists, community members in general, and scientists that help us to conserve the natural world. Now, one of the reasons we were initially founded 26 years ago to help Great Smoky Mountains National Park is the fact that Great Smoky Mountains National Park is wildly diverse in biology. It has a wild amount of biodiversity, and there's lots of reasons for that. Some of those reasons that Dr. O'Connell might be speaking about today is the topography. It varies greatly in terms of height of some of these mountains. It's also related to some of the geology. There's ancient geology in these Appalachian Mountains, as many of you know, and also the fact that there's a lot of water in Great Smoky Mountains National Park. It is a temperate rainforest, which means that it receives over 55 inches annually of rainfall. And so all of this together makes Great Smoky Mountains National Park wildly biodiverse. But within that, of course, there are always threats to that same biodiversity. And so there's a lot of information that we need to keep intact and understand better in order to conserve some of these species that we see. And one of the things that our executive director at Discover Life in America, Todd Witcher, says regularly is that we can't protect a species if we don't know that it exists. And so it's really important that we understand what organisms are all throughout Great Smoky Mountains National Park and how those organisms interact. This little guy is a robber fly. It's got a couple great common names, like the gnat ogre or the dusky goggle eye. But as you can see, this little guy has great vision and is still being studied by a number of scientists to better understand how that vision works and why it's able to be so accurate when it goes after its prey of fruit flies and other small insects. This is just one example of one organism that we can find in Great Smoky Mountains National Park. And so the key for us at Great Smoky Mountains National Park and Discover Life in America's kind of flagship project is something called the All Taxa Biodiversity Inventory. And it's quite literally inventorying every single species in Great Smoky Mountains National Park and understanding who's there. But not only understanding who's there, also understanding how common they are, where they live, and also how they interact with one another. And having all of this information is really important, not only for the park, but for the greater world, wherein we can start to piece together some of the relationships that these organisms have, how much they rely on one another. And within that, we can start to understand the impacts that we as humans have if one small species not only goes extinct, but if we cut it down in this one specific area of the world, how is that impacting the entire web of biodiversity that we see? So Discover Life in America, since its founding 26 years ago, we've determined that there are over 22,000 known species in Great Smoky Mountains National Park alone. Within that, we have over half of those completely new records for the park since the founding of Discover Life in America and the beginning of our All Taxa Biodiversity Inventory. And also within that number, something to be very proud of, over 1,000 of those 22,000 known species are completely new to science. And so a lot of that we have to thank Discover Life in America for in this All Taxa Biodiversity Inventory, helping us look for the small things and understanding what organisms are present. Now within that 22,000 organisms, we have quite a few here. One thing that I like to point out is that our little sliver here with the bird in it, those are the vertebrates. Those are where you and I belong when we're in the park. And we've actually had to make this sliver slightly larger so you can actually see it. These are the big organisms that people often think about when they go to the Smokies, the bears and the elk, the deer, and it's amazing what a small amount that they contribute to the biodiversity as a whole. You can also see over here, just to the right of those, our microbial friends, which Sean will be talking about today, and lots of other different species that play large roles within the park itself. Something that we recently added to our park list. This is a fairy wasp, just as an example of things that we find here in the park. This fairy wasp is only 0.75 millimeters long. If you pull out a ruler and look at that, I think it will blow your mind a little bit. These are tiny insects, and they're so small that they actually have to have these very kind of feathery wings in order to keep themselves aloft and have control over their flight. So another example of some of the small things that you might find in the park if you remember to look close enough. Aside from the All Taxa Biodiversity Inventory, which again is our flagship project for Discover Life in America, we do a number of different things, including really important summer internships to bring about the newest group of biologists and microbiologists in the world. We have a podcast. We also do this
So I just lost sound on my end. Can you all hear me? Can somebody let me know in the chat if you can actually hear me speaking?
Yeah, Sean, we can hear you.
Okay, thanks. I'll go ahead. Sorry to miss the last couple slides that Sam just presented, but I'm very grateful to be here, and I thank Sam for inviting me and for Microbial Insights for hosting this webinar, and Marshall for helping to set it up. And I regret to inform Sam I have not yet made it to the Museum of the American House Cat, but that is on my to-do list. My kids will be happy to get there. So I'm going to go ahead. Sam introduced me and kind of where I started. I also wanted to acknowledge Sam's help in making much of the research results that I'm going to share with you here in a minute possible, and really greatly expanded what we've been able to do at Western Carolina University in terms of understanding the microbial diversity in Great Smoky Mountains National Park. I specifically like to also thank Will Kuhn and his interns from last summer, 2023, from Discover Life in America, who did much of the sampling work. So it was kind of a mixed emotions that we let them take over the sampling of the sites for us. We didn't get to go on the hikes and backpacking trips, but they did a great job in getting samples that we'll talk about here in just a minute. And over the years, next week we start my school here at WCU. It'll be my 24th year, starting to teach next year. But over the years, I've had a lot of support from in-house. So I just wanted to acknowledge up front some other groups and funding agencies that have made this work possible. So the biology department here and the graduate school, we have a master's program. So a lot of the results I'll show you were done by undergraduates as well as master's students. Up at Purchase Knob, the Appalachian Highlands Science Learning Center, we got a research grant recently to do some of this work. And then early on, Discover Life in America had a pretty robust research grant program, and we benefited greatly from having that support to make this foundational work be able to start and continue. Specifically, I'll point out a couple of recent graduate students that work with me, Elin Swank and Ivan Emmerich, who are going to basically, a lot of the results I'll show you were based on their work in the last 3 years. And then I also would like to point out Paul Super from Great Smoky Mountains National Park, who has been with me from the start. He gave me my first look at some of these sample sites in the Smokies, hiking down unmarked trails and finding these places, and then finding them again 20 years later to be able to do some of these projects. So what I'm going to do with y'all here in the next little bit is basically reiterate a little bit of what Sam said about what draws, I think, people to national parks and to understanding life in pretty interesting places. But then to go down to the level that I work with, which is, I call it the subvisible level, but the microbial organisms that really make all of the other organisms able to do what they do. And so I'll mention three projects that we've done over the years. The first 20 years or so, we're really continuing this on today. We've looked at trying to grow bacteria and archaea. I'll mention we haven't had any luck with the archaea yet in my lab, as well as look for organisms that are out there that we don't grow or can't grow readily right now, but we can still know that they're there, perhaps understand what they may be doing. And that is a lot of what I'll present in the other two projects too. So the second project will be the Microbial Insights work that really got us to explode the number of organisms that we're able to understand. And for the first time, look for fungi and nematodes along with these bacteria and archaea, the prokaryotes, the smaller organisms. And then lastly, thanks to Microbial Insights, we also were able to look at not only who is there, but maybe what they're doing. That ties together some of what Sam was talking about in terms of understanding first what species are present, but then importantly, how do they work together, and what are they doing maybe to help the environment along, or the habitats along in which they reside, or maybe how they're impacted by things like disturbance, including climate change. And then I'll just go through some brief conclusions. There's a lot of kind of Greek and Latin names in here, and I'm not going to go too heavy into that, but I do want to point out some of the big trends that I think we've picked up on over the last 20 plus years now. So as Sam said, really kind of these large organisms are what draw, I think, our eyes to national parks in particular. And I was fortunate enough to work in Idaho and be about an hour and a half's drive from Yellowstone National Park, which is also a hotbed of microbial ecology work back in the 1990s when we were first understanding extremophiles. But that national park has things like grizzly bears and wolves and mountain lions. And so the Smokies have some pretty charismatic megafauna too. And I just like this picture of a mother elk and her young fawn from last week that my daughter took. But if you go down to smaller scale from there, these are photos I took close to the Smokies. You can start to appreciate some of those small vertebrates Sam mentioned that are really the minority in terms of the diversity of life in the Smokies. But you can get down and look at things like passion flowers or hummingbird moths, maybe phlox, praying mantises, if you look closely. Where we go is even farther, and the title of my talk today, every fall I start with this quote in my microbiology course: the role of the infinitely small in nature is infinitely large. And that's attributed to Louis Pasteur. And I really like that quote because it does show that, yes, we're visual creatures. We relate to things on our scale and bigger and somewhat smaller. But if you go down even smaller, these microbes make the world go round. What's in the background of this photo is basically fluorescently tagged bacteria. So you can tell these kind of orange, yellow, globular cells, these longer red cells, some green and blue cells mixed in, etc. Each one of those is a different species. And really our limits to understanding microbial diversity is a limit of technology. So as we can get closer to the scale of microorganisms, we can start better understanding who they are and what they're doing, including in places like Great Smoky Mountains National Park. And I just point out one of the things I talk about in my classes a lot, which is carbon cycling. Essentially, it all comes down to carbon dioxide and then organic material, and microbes are the champion organisms for converting one form of carbon to another. They can get energy, such as in chemolithotrophic reactions, where they essentially can breathe things like metals in order to get energy. And so there's a lot of complexity to this, but one of the things that we think about in carbon cycling and carbon dioxide in particular is climate change and some answers to perhaps climate carbon capture technologies or whatever may reside in microbial applications. Okay, so they really are the drivers of ecosystems. And then I also like to show these kinds of introductory materials to my classes and to groups that I talk with. This is a cartoon version of what it might look like within soil, and most of what I'll talk about today is in soil environments in Great Smoky Mountains National Park. From the microbial perspective, so you can see sand, silt, and clay particles, little groups of microorganisms. They rarely exist alone. They're usually in these micro colonies, but you can see different species represented by different shapes and colors. And then the physical environment and chemical environment can be radically different, just a millimeter apart from particles. So you can have saturated areas with lots of water. You can have pockets of air, maybe different gases in that air bubble, organic matter or not organic matter. So really can drive how diverse these soil environments will be. And I like to just talk about these kinds of numbers. These are very small organisms in very small scale environments, but when you add them up, they really can account for huge amounts of material on Earth in processes. So people have estimated something like 10 to the 30th bacteria on Earth. So that's a 1 with 30 zeros after it. That's 9 orders of magnitude more than astronomers have estimated there are stars in the entire universe. So there's a huge number of microbes on Earth, bacteria primarily. About half of all organic carbon that's locked up in living organisms is in bacteria and archaea. And that makes sense, especially if you think about my past life working in subsurface environments. We can find living organisms a mile or 2 miles below the surface of the Earth and below the surface of the oceans. That adds up to a lot of biomass. And then this number is a little bit in play in terms of what it is, but probably greater than 50% of nitrogen and phosphorus in living organisms is also in microbial life. Okay. Within soil, one of the findings I'll show you that Microbial Insights helped us with actually confirms this number of about 1 billion cells per gram of soil. Think about a teaspoon's worth of soil, maybe 1 billion organisms in there. And those billion organisms, especially in some pristine environments like you might find in regions in Great Smoky Mountains National Park, perhaps millions of species within that gram of soil. So that's one of the things we sort of wrestle with contributing to the all taxa biodiversity inventory is what is a species, and then if we actually can get more species accounts, that number is really going to be a huge part of the slice of life in the Smokies. And then just a couple of things that I'll point out. All other organisms that are non-microbial rely on microbial symbionts or mutualist organisms that help them do things like digest food, keep away pathogens, say from soil roots in the soil for plants. And so it's been shown in human models and other animal models that of all the genes included in the host and their microbial inhabitants, 99% of those unique genes might actually be coming from the microbes, and some of those genes play a big part in the health of the host. And then I pointed this out already, but one of the things we're always curious about is how we can get more microbes into the lab in culture, meaning growing up by themselves on a petri dish or maybe in an aquatic kind of medium, but away from others. And it turns out that doesn't happen most of the time. It's just organisms like to grow together, and so separating them actually means that they won't grow. Just another introductory slide to show kind of my perspective on life on Earth. This is a fairly recent tree of life model from a textbook that I use in class that shows pretty much just how diverse a domain bacteria is. So there's 3 domains of life: bacteria, eukarya, and archaea, and much of the major diversity across life is within the bacteria. And that makes sense in part if you think about it. Bacteria were the first life forms to arise on Earth, and they've had 4 billion years maybe to expand to many niches and habitats, including on the eukaryotes and associated with some of the archaea. Archaea and eukarya are actually more closely related to each other than either is to bacteria, even though archaea under the microscope look very much like bacteria. And I won't say a lot about archaea today. We have done some work with them, but they are notoriously difficult to work with in non-extreme environments like from soil. So we haven't done as much as we'd like, but we're continuing some projects with them. But this work that Microbial Insights helped support in the last couple of years has allowed us to break into the eukaryotes in more detail. And I'll report on a little bit of fungi diversity data and then nematode data, which is one of the projects that Microbial Insights Discover Life in America really wanted to get at. So I'll mention those organisms in a little bit more detail. So just to segue into my first project that we've worked on at WCU, within soil, the diversity of life, understanding that diversity, who's there, is critical to understanding food webs, how organic matter gets decomposed and reconverted to other forms for life to continue to use. And then nutrient cycling process, you think about big things like nitrogen, carbon, sulfur. How does that move through ecosystems? So that often is within the soil habitats. And like I said, we've been working with archaea and bacteria, but we're looking at fungi and nematodes and trying to look at interactions there. One big, I think, take-home message thinking about size of organisms is the smaller in scale you go, the more diversity there is. And that's really the thing that confounds us with the all-taxa biodiversity inventory is, there's arguably millions of species in Great Smoky Mountains National Park just on the microbial scale. How do we get a grasp at that? And so I think this more recent work is allowing us to get further along by an order of magnitude more in terms of the diversity information. And then ultimately, like I mentioned a second ago, just understanding how these organisms interact. Really, again, that's limited by technology, and technology is getting better by the day in the microbial ecology world. Okay, so I just wanted to show this map of Great Smoky Mountains National Park without too much detail in terms of place names to represent what the park looks like in terms of land use history in part. And what's not shown directly is elevation changes, geology that underpins the soil types in many of these places, etc. But generally speaking, if you're not too familiar with the Smokies, the North Carolina-Tennessee state line runs kind of right through the middle. So Tennessee to the north, North Carolina to the south. And we're down here, Western Carolina University, south of Great Smoky Mountains National Park, about maybe 15 miles as the crow flies, but about a 40-minute drive to get in through the Oconaluftee entrance. And then you can go on up to Gatlinburg and beyond. But what we've focused on in these 20 plus years of working in the Smokies are established ATBI plots. So the work we started back in really 2002 borrowed heavily from work others had started within the few years before that in establishing plots that would represent vegetation differences across the park, which would also include elevational differences. Some are open grasslands or balds, unvegetated by trees, and others are more like you think about the Smokies being heavy forests, but of different kinds, coniferous or deciduous, cove type habitats, etc. So what I have outlined here in purple are the sites that we've done the most work in, especially most recently. And so Cades Cove, many people have heard of, I'm sure, and other places up high like Mount LeConte, up higher than Cades Cove, are the other sites we've hit. Albright Grove, which is an old growth forest up on the Tennessee side, we'll talk about a little bit in this talk. And then Cataloochee and Purchase Knob over on the eastern tip in the North Carolina side of the Smokies are some of our favorite sites to sample from. And these were ones that Microbial Insights helped us to focus in on for further analyses that I'll talk about today. Other sites are listed on the left side. These are not ATBI sites, but of interest. And so, for instance, Kephart Prong, just up inside the park from the Oconaluftee Visitor Center area, and the Oconaluftee Visitor Center area itself down here are sites that I've taken my classes to for many years and repeatedly sampled from streams and soils at those sites. So those have historic kind of interest from us and others. And then Paul Super from the Park Service, Keith Langdon, retired from the Park Service, Will Kuhn came up with these other sites that have been shown to have interesting insect life, plant life, etc., as possible places to look for interesting microbes. So these are also sites that we focused on recently with the support of Microbial Insights. Okay, so our first project, really when we started in 2002, was a brand new professor in the fall of 2001. I went to the Discover Life in America conference with my colleague Fred Coyle, who worked with spiders and other arachnids in the park. And he introduced me to a lot of people in Park Service, BLIA, and Keith Langdon pulled me inside and said, you really need to start working with microbes in the Smokies. And I said, okay. And so he pointed us to Albright Grove, which I just mentioned. And so what we did for a few years, lots of undergraduates and some graduate students and I, was sort of looked at diversity in soil in general, but also with some specific aims in mind. We were sampling from eastern hemlock stands just before the eastern hemlock Bobulia delgid started hitting the Great Smoky Mountains National Park and devastated a lot of those stands. So we wanted to get a baseline of microbial diversity along those hemlocks. The soils and waters at long-term study sites at Kaparkong and Okanalefty I mentioned. I won't talk much about the elk-associated bacteria in archaea, but we did basically track elk around and collected their fecal material to understand if we could identify elk based on their microbial symbionts. And we had some pretty good success with that in a master's and undergraduate project. And then before white nose syndrome arose or occurred in the park, we were able to get into one cave in particular, Gregory's Cave by Cades Cove, and sort of do some baseline microbial diversity work there. And so the methods that we use include growing microbes in culture or just getting DNA from soil and or fecal material, in this case of the elk, and looking at who was there without having any culturable representatives. And so what this looks like is ultimately the identity of these organisms, no matter what method we choose, including some of the ones I'll talk about later, come down to this molecule, ribosomal RNA. So all life on Earth, as that tree of life suggested, can be tied to the same branches based on having one molecule that every single life form needs, which is ribosomes to make proteins. And so the sequence of nucleic acid within that rRNA or adjacent related genes can give us that diversity information. And so we took two approaches early on. We would try to culture organisms. So this is just an agar petri dish with a pretty low nutrient load of foodstuffs in that agar. But what you can see is diversity that is pretty easy to distinguish from one another. So yellow colonies, sort of this concentric ring colony that's beige, some little purple ones, white, different sizes. So with our eyes, once we can get these single cells or small groups of cells to grow up to millions or billions of cells, we can actually then do something with them. So we can take them to a new plate, get them to grow alone if they'll do that, and then do things like look under the microscope to see what cell sizes, biochemical parameters they might have based on staining them, and then perhaps growing them and understanding what kinds of foodstuffs they might like. So most of this is going to be related to organisms that grow on organic carbon. Molecular methods, as I'm calling here, what we can do is basically just shred the cells open from the environmental matrix, extract the DNA from that soup, and then take a variety of tools to be able to look at this one gene by amplifying it with something like polymerase chain reaction, and then sift it out with some method like cloning, or another one that we used a lot was called DGGE. But basically you can separate each species perhaps as a band on a gel or a different colony of E. coli that's incorporated a new piece of DNA in cloning. But the DNA sequencing of that ribosomal gene is what we'll use to tell one organism from another. And at this point, getting DNA out of a culture is very straightforward, so we can just feed that right into that same process. And this early work, we had about 500 isolates and still have those in the freezer here at WCU. And we were able to get about 1,200 sequences, which you'll see in a minute is dwarfed by what we were able to do most recently. Okay, so this is another figure from the textbook that I really like and use for a couple of my classes. But what I want to do is just kind of orient you to some names that I'll kind of bounce off with you throughout the rest of my time, including at the phylum level of bacteria. And so the most diverse group of life on Earth, probably, but certainly within bacteria, is the phylum Proteobacteria. This blue wedge, and that width of the wedge suggests how much diversity is incorporated in there. And any of the groups that are shown here have at least one cultured representative, but this, I'll show you in a second, has a lot. We also have a group called the Bacteroidetes, that is a phylum, Actinobacteria, and Firmicutes. And these are 4 groups that are very easy to find across any environment on Earth, whether it's an animal host, soil, water. Typically, you'll find these 4. The ones that are not as easily found, at least in culture, are many of these other groups. And I won't go into all these names, but if you can think of Proteobacteria, Firmicutes, and then also dwell on Acidobacteria in a minute, those are some big ones in Smokies. This other part of this figure that I like a lot just shows on a log scale how many species are estimated to be out there for each of these groups. So Proteobacteria, for instance, is broken down into classes, 6 classes, but you can see it's almost 1 million species just of that Proteobacteria phylum. And the colors show the end on the red is how many are out there based on surveying genes from the environment, the 16S, the rRNA genes. The green bar is the how many of those total species have been cultured. And so you can see, again, log scale here, thousands of species of Proteobacteria cultured, but there's probably millions out there. So we're just at the small end of what we can culture. And I'll just point out one other group, since I'll bring about later. Acidobacteria is another one that's pretty diverse in terms of species richness, how many species are out there, maybe hundreds of thousands, but only hundreds of cultures. So there's a lot of work left to do in microbial ecology in the Smokies and elsewhere. And what I like to do, because there's just so much information here in a short time, is I won't go through all this, but if you look at just the diversity of the cultures that we got in, say, the first 15 years or so of our work in the Smokies, we're only picking up about 6 phyla. So there's Proteobacteria. The size of the word indicates how much richness of species we've gotten for those groups. And so Proteobacteria, Firmicutes are the 2 big ones. And then these guys, Acidobacteria, Deinococcus thermus, we've only actually gotten 1 or 2 cultures of those, whereas we've had hundreds of the other ones. And just to show some of the depth of at the genus level, which is really where a lot of bacterial records end up being kind of stopped, because we can't get down to species very easily for many organisms. But at the genus level, you can see some common names. So Painobacillus, a very common soil organism, happens to be in Firmicutes. Pseudomonas, common in waters and soils and associated with plants and animals, is Proteobacteria. So kind of focus your eyes again on the phylum here. I'll switch to what we found in the non-culture work, just extracting DNA from the environment and seeing who's there. And what you'll see is more than a doubling of the number of phyla that we can detect out there. And Proteobacteria is still common, but it's much smaller. Firmicutes is even less common based on this method. So the take-home message here is you're going to see different kinds of diversity with these 2 different methods. And so probably what's happening with Acidobacteria is they're very common in the environment, but they do not like to be cultured. Proteobacteria is also very common, but we kind of maybe over-culture them in the lab because they're readily able to grow on media. And then if you looked closely at these slides, I guess this will be recorded if you really want to come back and look at them or ask me for more information later. I'm happy to share that with you. Those kinds of names at the genus level are not overlapping very well at all with the culture work. So that was sort of our first 15 years of work or so in the Smokies. And what's jump-started us and really allowed a couple of my past master's students that have worked with me to greatly expand on our understanding of diversity has come about because of this gift from Microbial Insights that Discover Life in America managed. And so in this case, what we're using is methodology much like the DNA sequence-based method, but we're doing it at a high throughput level in Microbial Insights lab. Did that work for us. So we can come out with not just 1,000 sequences, but tens of thousands of sequences or more, and across many more habitats and environments in the park. The other thing, because Discover Life in America has this interest in understanding nematodes better in the park, is we used another method that Microbial Insights did, quantitative PCR, focusing in on certain nematode taxa that would allow us to look at diversity of nematodes across those many habitats. And so this is our first foray here at WCU into fungi and nematode diversity work. We're still kind of wrangling with the data and planning to get a couple of papers out the door, hopefully in the next few months, reporting on that. But we'd like to know how all of these groups interact. And so the last part of the talk, I'll come back to that. In the last part of this section, I'll also mention in passing. So what happened last year to make this work possible was Will Kuhn and his interns that I mentioned before, we trained them how to collect soil core samples at the Oconaluftee visitor area site by the river there, if you've been there, near where those elk were photographed, and basically took a soil core with a disinfected core. And previous to that, a former graduate student that kind of was a go-between between his thesis work and this work had already done some basically next generation sequencing, high throughput sequencing from 2 sites, Cataloochee and Purchase Knob. And in those sites in May, he was able to collect 3 samples from each case. What we decided to do for this work that Microbial Insights supported was try to capture not statistically relevant data within one site, but try to capture diversity across the entire park as much as we could, working with about 30 total samples. And so those samples were collected, contained in sterile Whirlpack bags, and most of the time were kept cold and then shipped up to Microbial Insights for analysis. The nematode data basically resulted from 53 targeted taxa that spanned basically lifestyles, whether the nematodes would feed on plants, bacteria, fungi, or more omnivorous tastes. And that was work done at Microbial Insights, as I mentioned. Fungi and bacterial data were generated with next generation sequencing. We had a different lab do the Cataloochee and Purchase Knob sites just because that predated this gift. So I'll cut to the chase and the results here, and then I'll break them down a little bit more detail for you. But of the 53 nematode taxa that were investigated, we found 40 across the park. And so at each site, we found some nematode diversity, 9 to 28 total taxa. And then there was 4 genera, these are at the genus level, that were found in every site that was sampled last year by Will and his team. Going to fungi, so dropping down a size scale here, we see increase in richness, so total species, or what we like to call OTUs. It's a bit of a cop-out in microbiology, but kind of roughly equate with species. 15 to 292, 300 or so species per site, so an order of magnitude perhaps up from the nematode. And then with the bacterial work, sequencing those pretty extensively, we got a minimum of about 1,200 species up to 3,300 species or so per site detected. So what does that look like? These are some photos that I found from a government site in Canada, just showing you what roundworms, aka nematodes, look like. So small organisms with maybe hundreds of cells that make up their bodies, but they would certainly have microbial partners working with them and some against them. And I'm just going to show you 2 slides, a little bit text heavy, but I think the highlights will jump out at you. So for the nematodes we found in the Smokies, this is based on the groupings that Microbial Insights provided. There are bactivorous groups and herbivorous groups that I'm showing on this first slide. And then in parentheses behind each of these names for a genus, if there is a genus name, there's not always, how many of the 22 sites that we sampled showed evidence for those nematodes being present. And so you can see range from 1 in some cases, Heterodera, up to 22, so Mesocriconema, for instance, from the herbivorous groups by site. And then what's, I think, most exciting for Discover Life in America and Great Smoky Mountains National Park is that the groups that are not shaded are new records for the Smokies. So these are organisms that hadn't yet been detected in the Smokies. They've been there all along. Nobody's looked for them perhaps in a lot of detail. So we will be able to expand the database for nematodes. The second set of data shows some of the nematodes that basically are fungivorous, can eat fungi, and then omnivorous, maybe eat bacteria, fungi, archaea perhaps, what have you. And then others, maybe we don't know what they're working on for their metabolism, but they're common in many soil environments. And again, you can see a lot of unshaded groups on this slide, so more new records for the Smokies. And in some cases, look like they're pretty extensively distributed across the Smokies, like Phaceria, Labronema, Tylenocolumellus, for instance. And then just site by site, this is a lot of information, and it really, again, is most of the time just one sample taken at each site, so you have to take it with a grain of salt. You'll see Purchase Knob and Catalucci were included here, so we were able to get a fair amount of diversity from those 2 sites based on having triplicate samples. But even Twin Creeks, where we only had 1 sample, and Tremont, where we had 1 sample, seem to be pretty rich in nematode diversity, so more than a couple dozen species detected. And then some other sites, Snake Den Ridge, Cades Cove Gum Swamp, it's lower elevation in this case, the Cades Cove sample, lower richness. But again, if we were to up our samples, maybe this bar would be over here too. But nematodes are everywhere, and some species appear to be particularly widely distributed. Okay, going into fungi, another group which I don't have a lot of background in, but I'm coming up to speed on this major group of life, closely related to animals, are the fungi. And basically, what I'm going to show are just at the phylum level 5 main groups. And you'll notice Ascomycota is the biggest phylum represented here. And again, this is complex. This almost looks like a stained glass window. But what I really want you to look at is there's 15 sample sites represented here, and it really doesn't matter which site is which. But you'll notice that no 2 of those patterns really look the same. And down here, it describes what these colors mean, but the bar size within the main bar suggests the proportion of sequences recovered were attributed to a particular genus. In this case, Russula would be this large piece here. But what we found with the fungi work was that there is plenty of diversity, but at a particular site, it may be one genus or a couple genera that really drive all of the diversity within that site. Okay, and here are some names of some of the genera that we recovered for fungi. You can see Penicillium, you might be familiar with, very common genus across the park, and then some other ones, so mushroom forming ones like Amanita, also a name that you might be familiar with. And richness of fungi, in this case, having replicate samples, Purchase Knob and Catalucci, does seem to have upped our ability to detect many species, OTUs, so 250. And this is some work that Ivan Emmerich, I'll mention in a second. His hypothesis there is a lot of times fungi form mycelial networks in the soil, maybe even mushroom-like structures. And so when you're sampling with a coring device, you may be picking up more fungi than soil in some cases, or quite a bit of fungi compared to soil particles. And so they may be skewed, some of these sites, to one phylum or genus or a few based on that. Okay, but generally speaking, fungal diversity, kind of centered around 50 species or more within the Smokies. Albright Grove, the old growth site up in Tennessee, northeastern corner of the park, where there's a lot of hemlock historically, and some tulip poplar that are 500 years old, for instance, seems to have some good diversity there too. Okay, so the last group within this most recent project that I'll mention is our prime group that we work with here in my lab, are the bacteria. And again, there's so much information here, thousands of species potentially from each site. So I'm just showing a highlight of some of the major groups. And at the phylum level, I've already mentioned acidobacteria and proteobacteria. So those are the two biggest pieces of the pie here. But you'll notice a couple of other names I'm going to come back to here in just a minute. And unlike the fungal kind of patterns of diversity, the bacteria don't look super different from one another from site to site. This is 15 of those 22 sites that were sampled within last year. And you also notice that at the genus level, a lot of these do not have formal names. And those are the groups that are pretty much only known about through DNA sequences from the environment. So GP means group 2, 3, 4, 5. These are all within the acidobacteria phylum. So one of the most common phyla we detect in the smoky soils. But there's a lot of diversity hidden within phylum and within the genus, as I'll allude to here in a minute. And so for the OTUs by site for bacteria, you'll notice we're pretty much starting at 1,000 or plus as our minimum number of species. So think about fungi was about 50 or so. So, radically different numbers of species that we're having to sort of sort through within these environments. And what's to me kind of amazing is within the samples that Microbial Insights analyze, just one sample from each park, we're getting more diversity from the other lab that did the Purchase, Knob, and Catalucci sites, even though there were more samples processed. Okay, so that's a good, I think, more robust diversity levels of information from Microbial Insights. And then this last thing, this is something my student that worked with me, Ivan Emmerich, just graduated in May, put together, and he's working on this for a publication. It's really complicated. I could not recreate this graph if I wanted to. It takes a lot of basically working with bioinformatics tools, including handling massive amounts of DNA sequence information, running them through tools, and then processing that, running them through new tools. But what this is a co-occurrence network. And I'm showing it for bacteria and fungi in this slide, so you can look at two major groups of organisms at one time. Anything in an elliptic shape is bacteria, anything in a rectangular shape is a group of fungi, and the names don't always mean a lot at first glance, but it represents one sequence type that was common in the database, and anything displayed in this particular graph is highly statistically significant. So it's a reduction of the total amount of sequence data, trying to put together organisms and how they might interact. And how you can infer that are through these lines, the edges as they're called. So a blue line between two groups represents co-occurrence. That means if you find one in the sequence data, they're always going to be found with this other group. And so this is a fungus, Mordiarella. This is a group of bacteria, the Tenobacteriaceae, a family of bacteria in this case. If it's a red line, that means if you find a group, you won't find the other group that it's linked to. So mutually exclusive. Basically what this does is allows us to start forming hypotheses to test for these interactions. And then gray lines, again, represent probably some interaction, but we don't know what that interaction is. Blue, I think you can assume is a positive interaction, or at least a strong interaction. Okay, and the width indicates that interaction. So what Ivan did was the bacterial and fungal work here. These are different nodes by color scheme, and came up with some of these relationships. Okay, and we're working on that, but you can see that Mordiarella is common around this plot, and so that suggests that group of fungi interacts with bacteria in a major way in many of these sites in the Smokies. So the next step would be maybe try to culture some of these fungi and bacteria and see if we can get them to interact in the lab. Is one parasitizing the other? Are they growing together on some common substrate and sharing resources? We don't know. And then just a quick look at bacteria versus nematodes. A little trickier to put next generation sequence data together with qPCR data, but Ivan did a good job of that. And again, you can see some really strong relationships based on these wide blue lines over on the left. But For instance, Plectus is a nematode that eats bacteria, and so perhaps one of its favorite prey items is Rhodomicrobium. Again, that's something, a hypothesis that we can perhaps test in the future. Okay, so that was the first time we were able to analyze next generation sequencing data for Smoky samples, and I'm really grateful to Microbial Insights for that opportunity, and Discover Life in America for helping make that come to reality. What I'm going to end with is just a couple of slides. It's actually pretty complex data, but I like what we have here because it allows us to start putting together not just like what species might interact with one another, but what maybe they're doing in terms of nutrient cycling, or etc. And so this is just going to show Purchase Knob and Catalucci as a couple of sites that we worked together on, and then with a graduate student. And we had that high throughput DNA sequencing from a different lab, but we tied it together with different qPCR that Microbial Insights does for functional genes. And I'll show that to you in a second. But this graduate student, Elin Swank, was most interested in nitrogen cycling under red oak forests with a kind of a long-term look at how the microbes and the oak interact together and how that might affect climate change, or how climate change might affect forestry practices in North Carolina. So I'll just show you a very busy slide, the nitrogen cycle, but mainly if you think about it, nitrogen from the atmosphere N2 is fixed to ammonia, NH3, and then ammonia can be go through ammonia oxidation / nitrification to nitrite to nitrate, and then this oxic-anoxic interface can show different processes, say deeper in the soil or in pockets of soil without oxygen, where that nitrate can be denitrified back to N2. So again, we just have a couple of minutes, so I won't go into this in much detail. But what you'll see on the left here now is the data from Microbial Insights. And here is the count of total bacteria. These are most of the time averaged between 3 replicate samples from Purchase Knob in this kind of blue and Catalucci in this kind of orange. But their estimates from qPCR are billions of cells per gram of soil. So that's consistent with what I told you previously. And actually, it's the first time I've confirmed that number in my own lab, so that was cool. And then archaea, one of the reasons we don't see a lot of archaea, don't work with them other than them being hard to grow, is they're also numerically much less common in soils in places like the Smokies. So perhaps millions, tens of millions of cells per gram. But really the take-home message of this slide is if you look at just the genes that are involved in cycling nitrogen, I've drawn these purple lines to these different processes. Within this qPCR approach to these genes that actually do these processes to convert one form of nitrogen to another, we can find species, infer that there's species within these samples that are doing those processes from pretty much all aspects. And then if you look at this in one other way, which is, again, a kind of a busy slide, but the same information on the left, and just looking at the diversity of bacteria, Catalucci, Purchase Knob, at the phylum level, those genes plus the DNA sequencing efforts, we're actually able to tie most of these processes to some group on this bar chart here. So for instance, proteobacteria, it was so many groups represented from these genes pointing to it, I had to kind of spread them out. But the most diverse phylum of bacteria on Earth also shows the most diversity of functions within a sample site that we might be able to measure. And then acidobacteria, not as well understood, but quite diverse. You know, iron reduction is one possible process. So cycling of sulfur, iron, nitrogen, and then also methanogens. We do have some archaea signature in this qPCR that we wouldn't get easily, you know, thousands of gene copies or cells per gram of soil. Some of these can be involved in things like nitrogen fixation too, those species. So this is really put this together yesterday, trying to make sense of this, but this is work Elin Swank did here as a master's student, and we're working on trying to publish soon. So kind of getting a fresh look at it. So I'll end there. I just wanted to point out a couple of highlights. And basically, there's high diversity of bacteria in the Smokies. We can see that with whatever method we use, but with these more modern methods, next generation sequencing, we can really explode the kind of ceiling of what that total number of species might be. And going from bigger to smaller microbial life forms, you get more diversity. And so I just mentioned 4 of the common nematode groups. Fungi dominated by usually Ascomycota or Basidiomycota, and it seems to be sample from sample how that works. And if you remember no other words about bacteria from this presentation than acidobacteria and proteobacteria, you're in good shape. Acidobacteria, unlike many soil habitats across Earth, seem to be more dominant in the Smokies than proteobacteria. It's usually the other way around. So acidobacteria seems to be a really important phylum in the soils in the Smokies, and that's a group we're continuing to work on. Addition of other genes besides ribosomal genes can help us tie who's there to what's going on with things like nutrient cycling. And then this work is continuing, so I'm hoping this year, like I said, to get a couple of things out for publication, but also to share with DLIA and the Smokies some final lists of species based on pretty much most of this work over the years. So I'll stop there. I don't know if there's time for a question or two, but I'm happy to stick around for a minute or 10.
Thank you, Sean, for that. We
Sure.
might be running a bit tight on time as far as questions go. What I can do is if you have a question in the chat you've already placed, or if you have further questions, you can just go ahead and email info@microbe.com. And that way we can forward you any questions that we have, Sean. That way we are quite respectful of everybody's times. And I get a lot of ones in the chat saying, great presentation, thank you, and they all loved it. So once again, thank you so much.
Oh, happy to do it. Sorry, I went over a little bit.
Oh, you were totally good. Everybody is a big fan of the presentation. I'm certainly impressed on the work going on in the Smokies as well. If that's all you have to say, once again, thank you so much, and we'll be in touch with any questions that come in. A lot of thank yous coming in. And that'll be it for today, folks.
Okay, well, thank you so much for having me. I appreciate the time to talk about this work.
Of course, thank you.